Hello everyone, so today we have the part 2 of the crime scene investigation PCR lab, and we start with the preparation of 1x TAE buffer. So the stock one is 50x and you need to prepare 2 liters of 1x TAE buffer. So here in your lab book you will need to calculate and document the volumes. Then you will be using this 1x TAE buffer for the gel preparation. So you will be casting gels with the agarose dissolved in this TAE buffer. And also it is going to be used as a running buffer for gel electrophoresis. Okay, so the next what you do, you cast the gels. First of all, of course, you weigh 1 gram of agar or agarose, and it is going to give you 1% gel. So you weigh it, then you put it in the Erlenmeyer flask and bring to volume of 100 milliliters. Of course, you dissolve it in TAE buffer. But the thing is, the agarose is not easy to dissolve, that's why you're going to heat it. And heating is going to be performed in the microwave. You could also use the heaters, but it takes much more time. So for the microwave, you're going to use the maximum power, and the time is 1 minute 30 seconds. So in this case, every 30 seconds you're going to check your gels, and you don't want to boil it, you just want to dissolve it, okay? So every 30 seconds stop the microwave and check the solution. When it's totally transparent, you can remove it from the microwave and let it cool down on the bench. While it's cooling down, you can assemble the forms in which you are going to cast the gel. Then, after we obtain about 45 degrees of Celsius, we can slowly pour the gel or the solution of the gel into the form, and you want to avoid the formation of bubbles, because if you take the gel, so let's say we have 3 wells here, and at some place you will have a bubble. When your DNA is applied the current, so minus and plus here, it will move towards the plus pole, and if it encounters the bubble, it will kind of try to go around it. And of course, in this case, you will not receive the clear result. So let's say that we have 2 parts, one was going straight, another one was going through this bubble, and in the result you will obtain something like that instead of a good band like this. Okay, so you leave it at room temperature to solidify. In the video you will see 2 types of gels that we're going to use. One type is usual, and we're going to stain it with fast blast dye. The second one is actually fluorescent, and fluorescence is given by the cyber green fluorophore, cyber green. So what happens here, the cyber green is positively charged, and in this case it will move from plus to minus, right? And whenever it encounters the DNA, it will form a distinct band which will be visible under the excitation with UV light. So we will be using UV light, okay? Okay, so we have the next step of the video series, and on this step we are going to prepare the samples for loading on the gel. So you are given the 5x loading die, and also you are given the DNA ladder. So what is ladder? Usually it is the set of DNA fragments of the distinct sizes. So let's say that we have inside this DNA ladder we have fragments of 5 kilobase, 2.5 kilobase, then 1.2 kilobase, and 1 kilobase. Okay, so this on your gel you will see as the ladder, and because of the presence of this DNA ladder on your gel, you can easily compare your own bands. So let's say you have 2 bands here, and of course it will tell you that the band number 2 is bigger than 1.2 kilobases but smaller than 2.5 kilobases, as well as the band 1 is bigger than 2.5 but smaller than 5 kilobases. So this is about the DNA ladder. And the loading die, again, it is using the glycerol to make sure that your DNA stays in the well and does not float away from the well. So now what you need to do, you need to calculate the amount of loading dye that you need to add to your 40 microliter samples, and after you calculate it, you document it in your lab book. So you add the loading dye to your samples, and then you are spin down the samples. Why would you need to do that? Because you have, let's say, the PCR tube, and your sample is on one wall and the loading dye is on another wall. Okay, so sample here and loading dye on this part. So to mix them in the first place, you will need to have them in the same place. That's why you are going to spin them down and obtain the mixture of sample and loading dye. So in the fourth video you will see how to load the gels with the samples, and first of all you are going to pour 1x TAE run-in buffer into the gel tray. So usually it takes about 1 liter, and this run-in buffer should cover your gel and also fill the wells in your gel. Okay, so what you do when you load it, first of all you take the sample and everything is spun down. Next you are going to place the tip of your pipette inside this solution and pipette it up and down so that you mix it well again. Okay, so after that you're going to take 20 to 25 microliters and place them in the well. So let's say this is a well of your gel. Okay, so here should be the level of your run-in buffer, and you should place a tip of the pipette inside the well. If you are putting it above it, the part of it will just float away. So don't do that. Try to really dip it in the well and then slowly release it. You push on the button on your pipette, which creates the pressure, so the samples will be released inside the well. Okay, do not release this button before you remove the whole pipette tip from the well, otherwise you will suck up the sample back, and also you will create the vortex here, which will lead to the floating of your samples again. Also, why you would need to do it slowly, because if you make it with a huge pressure, the sample will counterheat the bottom of the well and float away. Okay, another word for the loading dye. So why do we use the loading dye? In the video I said that we really need to bind the DNA to glycerol, And of course, if we don't use the loading die, the DNA will easily float away here. Okay, so be careful and use all of these tips. Of course, you can use your second hand as a block so that this tip will not be shaking, and of course it creates more precise conditions for your experiment. After you loaded all the wells, you will need to close the lid, and of course you have this tray here with two electrodes. One will be red, the second will be black. Black is usually for negative and red for positive. You should connect them color to color, so red to red and black to black. On the video you will see the four columns of the connections or connectors, and you will need to use these columns, okay, because they are related to the same output. If you plugged red to this connector, you should plug the black to the same column connector of the black. So this is the right way to connect. If you want to use red here and black here, it won't work, and you will see the error message on your screen. Of course, the wells of your gels should be closer to the black pole. Why? Because the DNA is negatively charged and it will move towards the positive electrode. If you connect black to red and red to black, you will have positive here and negative below. In this case, from your well, so let's say this is your gel, and here is the loaded well. Of course, the DNA will easily go through this part and you will lose the experiment, so be careful with the connections. Black, which is negative, is on the top of the gel tray, and you connect it to the negative connector. Okay, so after you connected everything, you should set up the power supply, and we're using 120 volts. It is the constant voltage. Also, you could increase it to 130, but not more usually. You could also decrease it to 100, but in this case, of course, it takes much more time than 120 voltage. After that, you're going to wait until your bands travel on your gel about 2/3 of its length. If we wait more, we are in risk of losing some smaller DNA fragments. So usually 2/3 or even half of the length of the gel. In the next video you will see how to dilute the fast blast, and we are going to use 5x working solution. So for this staining I am preparing 500 milliliters, and it is our final volume. This is our final concentration, and the stock concentration is 500x. Now you need to calculate how much of fast blast stock you need to dissolve in how much of water. Of course, you document it in your logbook. So you collect the gels from the tray and rinse them in distilled water. And after that, you can pour the fast blast working solution, and you should cover your gels with the fast blast dye. Otherwise, they will easily dry out and you will not be able to take a picture of these gels. You usually put it on the agitator or the shaker, it is also called shaker, and leave it overnight. After you put it on this shaker, you should set the appropriate speed, because if it's too much, the fast blast will be spilled from your gel holders, and they will dry out, so be careful with the speed of the shaking. Okay, so this is the possible results that you are going to obtain. So on this part we have the fast blast dye, and here we have cyber green. Okay, so you can see the ladder here. You can see the crime scene, A, B, C, and D suspects. So as I said, we have the DNA ladder, which starts from above. So the highest band will show you the heaviest one. So knowing this distribution of the bands, you will need to identify the crime participant in the list of suspects. Okay, so here on this side you see the fast blast, and as well as in the DNA fingerprinting lab we don't see the distinct bands. So if you here can distinguish between these bands, so this is 1, 2, 3, 4, 5, 6, 7, 8. Here you more or less see the second, third, okay, so second and third bands. The rest are not shown very distinguishable, and also you can see the very faint heaviest band in the ladder line. Okay, so again we have the standard gel, and from one side to another we have 8 centimeters. So these 8 centimeters you're going to calculate. If you are needed to calculate, you're going to calculate the distance between the bands and identify, let's say that for CS you obtain the values that are very close to one of your suspect's values. So let's say suspect X. And by this rough equation, you're going to identify the person who really participated in this crime scene. Okay, so this is the end. I will upload these lab results on the Moodle, so you will be able to use either printed version or the image editors with the special tool called Ruler. Okay, thank you all for watching. Goodbye.